Hello, my name is Ian Sinkowski, and today I will be presenting to you on the bicolor damselfish. During this presentation, I will go over background information, fun facts, physical description, classification, courtship of the bicolor damselfish, courtship swim patterns, big takeaways from this experiment, multiple Schwann's mass in bicolor damselfish, and big takeaways from this experiment. Background information: its scientific name is Stegatus partitus. Its family is Parmenistridae. Subfamily Parenticena. Category damselfish. It can get up to 2 to 3.5 inches long. It can be found in depths of 20 to 80 feet, and its distribution is the Caribbean, Bahamas, Florida, Bermuda, and the Gulf of Mexico. Some fun facts about the bicolor damselfish is the bicolor damselfish are native to the western Atlantic Ocean and the Caribbean Sea. This organism can be found hiding in crevices in coral reefs and large rocks. The bicolor damselfish is also the most abundant member of their genus. The bicolor damselfish mainly prey on plankton, which is unusual because most other species of damselfish feed on only algae. The male bicolor damselfish will aggressively defend their territory, especially when they are guarding their unhatched eggs. Physical description: the bicolor damselfish has an elongated dorsal fin. Fish use this for sudden darting-like movements, and it also aids in balancing. Caudal fins are also known as tail fins. Anal fins are on the bottom surface of the fish behind the anus. Fish use these for stability in swimming. Pectoral fins are located on each side of the fish around where the head meets the body. These are often thought as the fish's arms. Pelvic fins are located on each side of the fish near its bottom middle. These help the fish go up and down as well as turn sharply in the water. The bicolor damselfish is generally made of 3 colors: black, yellow, and white. In some cases, the yellow is less prominent. Classifications: the bicolor damselfish is in the kingdom Animalia, phylum Chordata, subphylum Vertebrata, superclass Gymnostomata, class Entinopetrigi, order Permissiformes, suborder Libromoides, family Pomecentridae, genus Stegastes, species Stegastes partitus. Courtship of the bicolor damselfish. In the study by Spears 2020, they tested the discrimination of the damselfish between different species of damselfish with recorded courtship calls. During this study, females did not respond well to the mating calls and therefore were excluded from the study. The standard courtship call would consist of 3 pulse chirps and is created by the adult male of all these sympatric species. While the damselfish chirps, it simultaneously swims in a courtship pattern termed the dip. Courtship swim patterns. The two most common movements in the patterns of courtship for the male bicolored damselfish are the picture you see up top, the male shows the coloration in his scales, and B, the dip, the male showing also this coloration in his scales. This type of tactic is very common in sea and land animals. Many animals try to flaunt their coloration in bright colors to attract a mate, such as the peacock. Some big takeaways from this experiment are males of the field and laboratory colonies of the bicolored damselfish showed a greater response to playback of courtship sounds of conspecifics than to playbacks of structurally similar and fully analogous sounds of the two closely related sympatric congeneric. The responsiveness by these groups differed. However, in the laboratory, males showed a significant difference in courtship during playback periods of congeneric sounds recorded in the laboratory, whereas field males showed no difference. Field males, though responding well to typical laboratory sounds from conspecifics during the tests, which did not include playback of field sounds from conspecifics, failed to respond well when playback periods did not include such field sounds. Big takeaways continued from this experiment. Present evidence demonstrates that male bicolor possess an excellent demonstrative ability within the acoustic modality, and such discrimination is based on some factors of structural patterning in time of the experiment sound. Present evidence also indicates that the observed response differentials noted during experiments were the result of slight but important differences in at least one typical parameter, the pulse interval. Multiple schwannomas in the bicolored damselfish. The bicolored damselfish in South Florida exhibit schwannomas, which are tumors that develop in the dermal layers but originate from the peripheral nerves and are an overgrowth of schwannomas cells. They can be internal masses taking up huge areas in the internal cavity of the fish. This can cause damage to the fish's organs and nervous system. This is especially important to the nervous system because damage to the nervous system inhibits muscle reflexes. The research suggests an infectious cause in the natural population and are investigating the transfer to healthy fish for study. If you look at the picture to the right, you can see two arrows where there are large schwannomas tumors exhibiting from the fish. You can also see on the bottom arrow that the area of the fish is extra swollen, which indicates that it could be damaging internal organs within the fish. Relevance to the medical field. This disease found on the bicolored damselfish has been compared to Rickerson's neurofibrosis color-changing spot nodules, which are found in humans. The spots appear to be the first sign of the disease and are recognized as schwannomas cancer cells. The bicolored damselfish is a small but hardy species that would be excellent to work in a laboratory setting. The bicolored damselfish would also be useful in research of the causes of benign and malignant nerve sheath tumors. The bicolored damselfish also provides value to epidemiologic studies on factors associated with occurrence of these tumors in small populations. Big takeaways from this experiment continued. The bicolor damselfish is the first clear-cut example of a persistent and widespread occurrence of such tumors in a natural population. Damselfish with multiple schwanns could be useful models for investigating the development of patterns of invasion, morphological cells variation. These are some of the references I used for this presentation. These are the last of my references. I would like to thank you all for listening, and I hope you learned something today.